So we are starting the chapter 4, that is principle of inheritance and variation. So let's start the chapter. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents? Or do they show some differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? This and several related questions are dealt with scientifically in a branch of a biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well-known Indian breeds. Example, Sahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of this phenomena. Understood? Next, we are going to read Mendel's laws of inheritance. It was during the mid-19th century that headway was made in the understanding of the inheritance. Gregor Mendel conducted hybridization experiment on a garden piece for 7 years. कौन से ईयर से कौन से ईयर तक? 1856 से 1863 तक. 1856 से 1863 तक, and proposed the laws of inheritance in living organisms. During Mendel's investigation into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiment had a large sampling size which have greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generation of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits. Example, tall or dwarf plant, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination, cross-pollination experiment using several true breeding pea lines. A true breeding line is one that have undergone continuous self-pollination. यहाँ पे चेंज करके क्रॉस पॉलिनेशन कर सकते हैं, बट करेक्ट है सेल्फ पॉलिनेशन. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated that is full or constricted green or yellow pods, and tall or dwarf plants. ठीक है, in figure 4.1, that that is table 4.1, it is given here. Okay, उससे पहले ये डायग्राम बता देते हैं, ठीक है। सीड शेप में राउंड डोमिनेंट है और रिंकल्ड रेसेसिव है, ठीक है। सीड कलर में येलो डोमिनेंट ट्रेट है, ग्रीन रेसेसिव ट्रेट है। फ्लावर कलर में वॉयलेट डोमिनेंट ट्रेट है और वाइट रेसेसिव ट्रेट है। पॉट शेप में फुल डोमिनेंट ट्रेट है और कंस्ट्रिक्टेड रेसेसिव ट्रेट है। पॉट कलर में ग्रीन डोमिनेंट ट्रेट है और येलो रेसेसिव ट्रेट है। फ्लावर पोजीशन में एक्सियल डोमिनेंट है और टर्मिनल रेसेसिव है। स्टेम हाइट में टॉल डोमिनेंट है और रेसेसिव है ड्वार्फ। क्लियर? अंडरस्टूड? 7 pairs of contrasting traits in pea plant studied by Mendel, जो अपन ने अभी पढ़ा है, ठीक है। नेक्स्ट टेबल में दिया हुआ है कॉन्ट्रास्टिंग ट्रेट्स स्टडीड बाय मेंडल इन पी, जिसमें कैरेक्टर है स्टेम हाइट, कॉन्ट्रास्टिंग ट्रेट है टॉल और ड्वार्फ, कैरेक्टर है फ्लावर कलर, कॉन्ट्रास्टिंग ट्रेट है वॉयलेट वाइट, फ्लावर पोजीशन एक्सेल टर्मिनल, पॉट शेप इन्फ्लेटेड कंस्ट्रिक्टेड, पॉट कलर ग्रीन येलो, सीड शेप राउंड रिंकल्ड, सीड कलर येलो और ग्रीन। Understood? Next is inheritance of 1 gene. Inheritance of 1 gene. Let us take an example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plant to study the inheritance of 1 gene. To study the inheritance of 1 gene, he collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial progeny or the F1. Mendel observed that all the F1 progeny plants were tall, like one of its parent, none were dwarf. He made the similar observations for the other pairs of traits. He found that F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial second generation some of the offspring were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plant, while the proportion of the plant that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. The tall and dwarf trait were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf. None were of in between the height. That is, all the offspring were either tall or dwarf. None were of in between height. Similar results were obtained with the other trait that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 stages, both the traits were expressed in the proportion that is 3:1. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 stages, both the traits were expressed in the proportion 3:1. The contrasting trait did not show any blending at either F1 or F2 stage. This is important. Okay. Based on this observation, Mendel proposed that something was being stably passed down, unchanged, from parent to offspring through the gametes over successive generation. He called this thing as factors. Now we call them as gene. उसने क्या बोला था? फैक्टर, और हम क्या बोलते हैं उनको? जीन्स. जीन्स therefore are the units of inheritance. कौन है units of inheritance की? जीन्स. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different form of the same gene. Slightly different form of the same genes. चेंज करके डिफरेंट कर देगा यहाँ पे, डिफरेंट फॉर्म ऑफ डिफरेंट जीन कर सकता है, बट करेक्ट है दे आर स्लाइटली डिफरेंट फॉर्म ऑफ द सेम जीन. Next is, if we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of the height, T is used for the tall trait and small t for the dwarf, and capital T and small t are the alleles of each other. Hence, in plants, the pair of alleles for height would be TT, that is capital TT, capital T or small t, or double small t. Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous. That is capital TT and small tt respectively. जिसे डबल कैपिटल T जिसे कहते हैं होमोजाइगस, और डबल स्मॉल टी उसे भी होमोजाइगस कहते हैं रेस्पेक्टिवली. ठीक है, डबल कैपिटल टी एंड डबल स्मॉल टी आर कॉल्ड द जीनोटाइप्स of the plant, while the descriptive terms tall and dwarf are the phenotypes. What then would be the phenotype of a plant that had a genotype capital T और small t? उसका होगा, क्या कहेंगे उसको, टॉल ही कहेंगे उसको. ठीक है, उसको क्या कहेंगे, टॉल कहेंगे. ठीक है. Next is, as Mendel found the phenotype of the F1 heterozygote, that is capital T small t, to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, as in the F1, and hence is called the dominant factor, while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other characters, traits, pairs that he studied. It is convenient and logical to use the capital and the lower case of an alphabetically symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and small d for dwarf, because you will find it difficult to remember whether T and D are alleles of the same gene character or not. ठीक है, confusion करेगा. Alleles can be similar as in the case of homozygotes, that is capital T and small t, or can be dissimilar as in the case of heterozygote, capital T or small t. ठीक है. Since the T, capital T and small t plant is heterozygote for the genes controlling one character, that is height, it is a monohybrid, and the cross between capital TT and small tt is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other, and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% chances of a gamete containing either allele, as has been verified by the results of the crossings. In this way, the gametes of the tall, that is capital T plants have the allele T, and the gametes of the dwarf small t plants have the allele small t. During fertilization, the two alleles T from one parent and through the pollen, and the small t from the other parent, then through the egg, are united to produce zygote that have one capital T allele and one small t allele. Clear? In other words, the hybrid have capital T small t. Since these hybrid contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygote, the F1 and the F2 plants can be understood from a diagram called Punnett square, as shown in the figure. It was developed by a British genetist. That is Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and left columns. All possible combinations are represented in boxes below in the squares, where all possible combinations are represented in boxes below in the squares, which generates a square output form. यहाँ पे डायग्राम दिया हुआ है प्यूनेट स्क्वायर यूज टू अंडरस्टैंड टिपिकल मोनोहाइब्रिड क्रॉस कंडक्टेड बाय मैंडल बिटवीन ट्रू ब्रीडिंग टॉल प्लांट एंड ट्रू ब्रीडिंग ड्वार्फ प्लांट. ठीक है, जिसका यहाँ पे रेशियो दिया हुआ है फिनोटाइपिक रेशियो टॉल 3:2 ड्वार्फ 1. जीनोटाइपिक रेशियो है 1:2:1, जिसमें कैपिटल टी स्मॉल टी ये है. ठीक है डायग्राम. ठीक है, आई होप समझ में आ रहा होगा. ठीक है, द प्यूनेट स्क्वायर शोज द पेरेंटल टोल दैट इज कैपिटल टी मेल एंड द ड्वार्फ स्मॉल टी दैट इज फीमेल प्लांट्स. द गैमेट प्रोड्यूस्ड बाय देम एंड द F1 capital T small t progeny, the F1 plants of the genotype capital T small t are self-pollinated. The symbol राउंड और उसके नीचे प्लस एंड राउंड और उसके ऊपर एरो are used to denote the female eggs and male pollen of the F1 generation respectively. The F1 plant of the genotype capital T small t when self-pollinated produces gametes of the genotype capital T and the small t in equal proportion. When fertilization takes place, the pollen grain of genotype capital T have a 50% chances to pollinate eggs of the genotype capital T as well as of the genotype small t. Also, pollen grain of the genotype small t have a 50% chances of pollinating egg of genotype capital T as well as genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotypes capital T, small t or small t. Clear? From the Punnett square, it is easily seen that 1/4 of the random fertilization lead to capital T, that is 1:2 lead to capital T small t and 1/4 to small t. Two, the F1 have a genotype of capital T small t, but the phenotypic character seen is tall. At F2, that is 3/4 of the plants are tall, where some of them capital T, while other are capital T small t. Externally, it is not possible to distinguish between the plants with the genotype, that is capital T and capital T small t. Hence, within the genotypic pair capital T small t, only one character capital T tall is expressed. Hence, the character T or tall is said to be dominant over the other alleles or dwarf characters. It is thus due to this dominance of one character over the other that all the F1 are tall through the genotype is capital T small t, and in the F2, that is 3/4 of the plant are tall through genotypically 1/2 are capital T small t and only 1/4 are capital T. This leads to a phenotypic ratio of 3:4. This leads to the phenotypic ratio of 3/4. Tall is 1/4 capital T plus 1/2 capital T small t, and 1/4 small t, that is a 3:1 ratio but a genotypic ratio of 1:2:1. The 1/4 is to 1:2 is to the 1/4 is to 1/2 is to 1/4 ratio of tt, that is capital T and capital T small t and small t is a mathematical condensable to the form of binomial expression, that is ax plus by square, that has the gametes bearing genes capital T or small t in equal frequency of 1/2. The expression is expanded as given below. That is, expression is 1/2 is capital T plus 1/2 is small t. अब is equal to 1/2 is capital T plus 1/2 is small t. फिर इंटू करके 1/t 1/2 capital T plus 1/2 small t is equal to 1/4 capital T and 1/2 is capital T small t plus 1/4 is small t. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generation. He concluded that genotype of the dwarf was homozygous, that is small t. What do you think have got had he self-pollinated a tall F2 plant? He concluded that the genotype of the dwarf was homozygous, small t. What do you think he would have got had he self-pollinated a tall F2 plants? From the preceding paragraph, it is clear that the genotypic ratio can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait. It is not possible to know the genotypic composition. This is for example, whether the tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. ठीक है, पहले का जो पढ़ा ना इसमें उतना इम्पोर्टेंट नहीं है, बोरिंग कर रहा था, ठीक है, स्किप कर सकते हो उसको, ठीक है, नींद आ रही होगी. So I am repeating it again. This is for example whether a tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. क्या किया मेंडल ने? टॉल प्लांट F2 का, ठीक है, क्रॉस कर दिया ड्वार्फ प्लांट के साथ. दिस इज कॉल्ड टेस्ट क्रॉस. टेस्ट किया, ठीक है. इन ए टिपिकल टेस्ट क्रॉस an organism P plant here showing a dominant phenotype and whose genotype is to be determined is crossed with recessive parent instead of self crossing. क्या किया? टेस्ट क्रॉस. an organism P plant here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self crossing. The progenies of such a cross can easily be analyzed to predict the genotypes of the test organism. Figure 4.5 shows the result of a typical test cross, where violet color flower, that is capital W, is dominant over white color flower, that is small w. Using a Punnett square, try to find out the nature of the offspring of a test cross. What ratio did you get? Using the genotype of this cross, can you give a general definition for a test cross? Can you give a general definition of a test cross? चलो मैं बताता हूँ तुम्हें जनरल डेफिनेशन ऑफ टेस्ट क्रॉस. सुनो तो टेस्ट क्रॉस की जनरल डेफिनेशन. A test cross is a genetic technique that determines an organism genotype by crossing it with an organism that has a non-recessive genotype. किसके साथ क्रॉस करके? An organism that has non-recessive genotype, जिसका अपने को पता हो, ठीक है. यहाँ पर डायग्राम दिया हुआ है 4.5 में. होमोजाइगस रिसेसिव जिसका रिजल्ट है ऑल फ्लावर्स आर वायलेट, और इंटरप्रिटेशन है अननोन फ्लावर इज होमोजाइगस डोमिनेंट, और होमोजाइगस रिसेसिव जो सेकंड वाला है उसमें दिया हुआ है रिजल्ट है उसमें हाफ ऑफ द फ्लावर आर वायलेट एंड हाफ ऑफ द फ्लावर्स आर वाइट, इंटरप्रिटेशन है अननोन फ्लावर इज हेट्रोजाइगस। ठीक है, ये दिया हुआ है उसमें। ठीक है, अंडरस्टूड। ठीक है, चलो, बेस्ड ऑन हिज ऑब्जर्वेशन ऑन मोनोहाइब्रिड क्रॉसेस, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principle or laws of inheritance. The first law or law of dominance and the second law or law of segregation. First law को कहते हैं law of dominance और second law को कहते हैं law of segregation. We are starting first law, that is law of dominance. First point, characters are controlled by discrete units called dash. करो dash को फिल, dash में आएगा factors. Characters are controlled by discrete units called factors. Second point is factors occur in pair. Factors occur in pairs. Third point is, in a dissimilar pair of factors, one member of the pair dominates, that is dominate the other, that is recessive. The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. ठीक है, द लॉ ऑफ डोमिनेंस इज यूज्ड टू एक्सप्लेन एक्सप्रेशन ऑफ ओनली वन ऑफ द पैरेंटल कैरेक्टर इन अ मोनोहाइब्रिड क्रॉस in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. Next is law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. Next is incomplete dominance. ठीक है, when experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, that is snapdragon or antirrhinum species, is a good example to understand incomplete dominance. ठीक है, the inheritance of flower color in the dog flower, that is snapdragon or antirrhinum species, is a good example to understand incomplete dominance. In a cross between true breeding red flowered R and true breeding white flowered plants small r, the F1 capital R or small r was pink. When the F1 was self-pollinated, the F2 resulted in the following ratio, that is 1 capital R that is red is to 2 capital R small r that is pink is to 1 that is small r जिसे white कहते हैं. ठीक है, here the genotype ratio were exactly as we would expect in any Mendelian monohybrid cross. Here the genotypic ratio were am repeating it again. Here, the genotypic ratio were exactly as we would expect in any Mendelian monohybrid cross, but the phenotypic ratio had changed from 3:1 dominant is to recessive ratio. But the phenotypic ratio had changed from the 3:1 dominant is to recessive ratio. What happened was that capital R was not completely dominant over the small r, and this made it possible to distinguish capital R, small r as pink from capital R red, and small r white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, a pair of alleles. In a diploid organism, there are two copies of each gene, that is, a pair of alleles. Now, these two alleles need not always be identical, as in heterozygote. Now, these two alleles need not always be identical as in heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modifies the information that particular alleles contains. Okay? One of them may be different due to some changes that it is undergone, which modifies the information that particular allele contains. Let us take an example of a gene that contains the information for producing an enzyme. उसके पहले ये डायग्राम दिया हुआ, डायग्राम बता देते हैं. ठीक है, इसमें दिया हुआ है क्रॉस दिया हुआ है. ठीक है, जिसमें ऊपर है कैपिटल आर रेड और स्मॉल आर वाइट. क्रॉस किया, ऑल पिंक आया F1 जनरेशन में, कैपिटल आर और स्मॉल आर. ठीक है, फिर उसको F2 में जनरेशन में फिनोटाइपिक रेशियो आया, रेड इज टू पिंक इज टू वाइट, जिसका रेशियो है 1:2:1 फिनोटाइपिक रेशियो. और जीनोटाइपिक रेशियो भी 1:2:1 है, जिसका रेशियो है कैपिटल आर 1, कैपिटल आर 2:1, स्मॉल आर 1. ये जीनोटाइपिक रेशियो है. ठीक है. रिजल्ट ऑफ मोनोहाइब्रिड क्रॉस इन द प्लांट स्नैपड्रैगन वेयर वन एलील इज इनकम्प्लीटली डोमिनेट ओवर द अदर एलील. ठीक है. Let's take an example of a gene that contains the information for producing an enzyme. Now, there are two copies of this gene, the two allelic forms. Let us assume, as is more common, that normal allele produces the normal enzyme that is needed for the transformation of the substrate. Theoretically, the modified allele could be responsible for production of the normal or less efficient enzyme. ठीक है, ये फर्स्ट पॉइंट था. सेकंड है ए नॉन फंक्शनल एंजाइम, और थर्ड नो एंजाइम एट ऑल. ठीक है. इन द फर्स्ट केस, that is first case is the normal less efficient enzyme. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotypic trait, that is, result in the transformation of the substrate S. ठीक है. Such equivalent allele pairs are very common. But if the alleles produces a non-functional enzyme or no enzyme, but if allele produces a non-functional enzyme or no enzyme, the phenotypic may be affected. The phenotypic trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represent the original phenotype, is the dominant allele. The unmodified functioning allele, which represent the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Again, I am repeating it. The unmodified functioning allele which represent the original phenotype is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Recessive trait किसकी वजह से दिखाई देती है? ड्यू टू नॉन फंक्शनल एंजाइम और बिकॉज नो एंजाइम इज प्रोड्यूस्ड. ठीक है, क्लियर. नेक्स्ट इज कोडोमिनेंस. ठीक है, एक बार और बता रहा हूँ वो पहले वाला. द अनमोडिफाइड फंक्शनिंग एलील विच रिप्रेजेंट द ओरिजिनल फिनोटाइप इज द डोमिनेंट एलील एंड द मॉडिफाइड एलील इज जनरली रेसेसिव. अनमोडिफाइड, ठीक है, डोमिनेंट है और मॉडिफाइड एलील रेसेसिव है. ठीक है, रिप्रेजेंट करता है. ठीक है, नेक्स्ट इज कोडोमिनेंस. टिल नाउ वी वेर डिस्कसिंग क्रॉसेस वेयर द F1 resembled either of the two parents, that is dominance, or was in between, that is incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example is different type of red blood cells that determine ABO blood grouping in human being. ABO blood group are controlled by the gene I, that is capital I. ठीक है, the plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene capital I has three alleles, that is IA, IB and small I. The alleles IA and IB produce a slightly different form of the sugar, while the allele I does not produce any sugar because human beings are diploid organisms. रीज़न है उसका, एलील जो आई है वो डज नॉट प्रोड्यूस एनी शुगर बिकॉज रीज़न है उसका, असर्शन रीज़न का क्वेश्चन का रीज़न है बिकॉज ह्यूमन्स आर डिप्लॉयड ऑर्गेनिज्म एंड ईच पर्सन possesses any of the two of the three I gene alleles. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over small I. In other words, when IA and I are present, only IA expresses because I does not produce any sugar. जब I बोल रहा हूँ मीन्स वो वाला स्मॉल आई है. ठीक है, सिंगल आई वाला. एंड व्हेन IB and I are present, only IB expresses. But when IA and IB are present together, they both expresses their own types of sugar. This is because of codominance. Hence, red blood cells have both A and B types of sugar. Since there are three different alleles. Since there are three different alleles. कितनी? Since there are three different alleles, there are 6 different combination of these three alleles that are possible. कितने? 6 different combination, and therefore a total of 6 different genotypes of the human ABO blood group given in the table 4.0. How many phenotypes are possible? कितने phenotype possible है बताओ. Answer is there are 4 possible phenotypes blood group for the 6 possible genotypes of the human ABO blood types. ठीक है, जो टेबल दी हुई है यहाँ पर, टेबल शोइंग द जेनेटिक बेसिस ऑफ द ब्लड ग्रुप इन ह्यूमन पॉपुलेशन. फर्स्ट कॉलम में एलील फ्रॉम पेरेंट 1, सेकंड में एलील फ्रॉम पेरेंट 2, थर्ड कॉलम में जीनोटाइप ऑफ ऑफस्प्रिंग, ब्लड टाइप ऑफ ऑफस्प्रिंग. सो जब IA पेरेंट 1 है और IA पेरेंट 2 है तो जीनोटाइप होगा ऑफस्प्रिंग का IA. ब्लड टाइप ऑफ ऑफस्प्रिंग है A. जब IA और IB होगा तो IA IB होगा जीनोटाइप स्प्रिंग का, ऑफ स्प्रिंग का, और ब्लड टाइप ऑफ ऑफस्प्रिंग AB. जब IA होगा और स्मॉल I होगा तो IA और स्मॉल I जीनोटाइप होगा और ब्लड ग्रुप होगा A. जब IB और IA होगा तो जीनोटाइप होगा ऑफ स्प्रिंग का IA IB और ब्लड टाइप होगा AB. जब एलील होगा IB और IB तो जीनोटाइप होगा IB IB, ब्लड टाइप होगा B. जब एलील पेरेंट 1 का होगा IB और पेरेंट 2 का होगा स्मॉल I तो जीनोटाइप होगा आई बी और स्मॉल आई, ब्लड टाइप होगा ऑफस्प्रिंग का बी। जब एलील पेरेंट का होगा आई और पेरेंट का भी होगा आई, दैट इज स्मॉल आई, तो जीनोटाइप होगा ऑफस्प्रिंग का स्मॉल आई और ब्लड टाइप होगा ओ। ठीक है, ओ कब होगी? जब सब आई 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 होंगे। डू यू रियलाइज दैट the example of ABO blood grouping also provides a good example of multiple alleles. Here you can see that there are more than two, that is 3 alleles governing the same character, since in an individual only 2 alleles can be present. Multiple alleles can be found only when population studies are made. Multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea plant is controlled by one gene. It has 2 alleles, that is capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grain are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, capital B seeds are round and the small b seeds are wrinkled. Heterozygotes produce round seeds, and so capital B seems to be the dominant allele, but the starch grains produced are of intermediate size in capital B small b seeds. So if starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. Starch grain synthesis है example, starch grain जो एग्जाम्पल है इनकम्प्लीट डोमिनेंस का। ठीक है, सो इफ स्टार्च ग्रेन साइज इज कंसिडर्ड एज फिनोटाइप, देन फ्रॉम दिस एंगल द alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. Again I am repeating it. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product, किस पे डिपेंड करता है? gene product, and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene. Clear? So the next is inheritance of 2 genes. Mendel also worked with and crossed pea plants that differed in 2 characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape, and one that had seeds of green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. Mendel found that seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. How can you tell which of the character in the pair yellow or green color and round and wrinkled shape was dominant? Here, can you tell which of the character in the pairs yellow and green color and round and wrinkled shape was dominant? Yellow seed color and round seed shape are dominant trait. ठीक है, सीड में येलो कलर जो है डोमिनेंट है और शेप में जो राउंड सीड शेप है वो डोमिनेंट ट्रेट है. ठीक है, thus yellow color was dominant over green and the round shape dominant over wrinkled. These results were identical to those that he got when he met separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols capital Y for dominant yellow seed color and small y for recessive green seed color. Capital R for round seed shaped seeds and small r for wrinkled seed shape. The genotype of the parent then can be written as capital R, capital R, capital Y, and small r, small r, small y, small y. The cross between the two plants can be written down as in figure 4.7, showing the genotype of the parent plants. The gametes capital R, capital Y, and small r, small y unite on fertilization to produce the F1 hybrid capital R, capital Y, small y. When Mendel self-hybridized the F1 plants, he found that 3/4 of the F2 plants had yellow seeds and 1/4 had green. He found that 3/4 of the F2 plant had yellow seeds and 1/4 had green. कब जब Mendel self-hybridized the F1 plants. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. ठीक है, यहाँ पे डायग्राम दिया हुआ है. ठीक है, मंडल का ये लो का. ठीक है, result of dihybrid cross where the two parent differ in two pair of contrasting traits, seed color and seed shape. पूरा एक पेज में डायग्राम दिया हुआ है. ठीक है, यहाँ पर जो फिनोटाइपिक रेशियो दिया हुआ है, राउंड येलो, राउंड ग्रीन, रिंकल्ड येलो, रिंकल्ड ग्रीन का है 9:3:3:1. राउंड येलो का है 9, राउंड ग्रीन का है 3, रिंकल्ड येलो का है 3, रिंकल्ड ग्रीन का है 1. ठीक है, नेक्स्ट इज लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. इन द डाइहाइब्रिड क्रॉस the phenotypic phenotypes round yellow, wrinkled yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination of series of 3 yellow, 1 green with 3 round:1 wrinkled. As a combination series of 3 yellow:1 green with 3 round:1 wrinkled. This derivation can be written as follow, that is bracket में 3 round:1 wrinkled, फिर bracket में 3 yellow:1 green is equal to 9 round yellow:3 wrinkled, comma yellow:3 round, green:1 wrinkled and green. कोई काम का नहीं है वैसे, बट पढ़ दिया. ठीक है, based upon such observation on dihybrid crosses between plants differing in two traits, Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. This law states that when two pair of traits are combined in a hybrid, segregation of one pair of trait is independent of the other pair of characters. This law states that when two pairs of the trait are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. Next is the Punnett square can be effectively used to understand the independent segregation of the two pairs of the gene during meiosis and the production of eggs and pollen in the F1. Capital R, small r, capital Y, small y plant. इतना इम्पोर्टेंट नहीं है जो कैपिटल आर स्मॉल आर बोल रहा हूँ. ठीक है, इसलिए जल्दी जल्दी पढ़ देता हूँ. ठीक है, कंसिडर द सेग्रीगेशन ऑफ वन पेयर ऑफ जीन्स कैपिटल आर एंड स्मॉल आर. 50% of the gametes have the gene capital R and the other 50% have small r. Now besides each gametes having either capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% capital R and 50% small r is independent from the segregation of 50% capital Y and 50% small y. Therefore, 50% of the small r bearing gametes has capital Y and the 50% has small y. Similarly, 50% of the capital R bearing gamete has capital Y and the other 50% has small y. Again I am repeating it. Therefore, 50% of small r bearing gametes has capital Y and the other 50% has small y. Similarly, 50% of the capital R bearing gametes has capital Y and the other 50% has small y. Thus, there are 4 genotypes of gametes, 4 types of pollen and 4 types of egg. The 4 types are capital R, capital Y, capital R, capital Y. Second is capital R, small y. Third is small r, capital Y. And fourth is small r, small y. Each with a frequency of 25% or 1/4 of the total gamete produced. When you write down the 4 types of egg and pollen on the 2 sides of the Punnett square, it is very easy to derive the composition of the zygote that give rise to the F2 plants. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Note them down in the format given. 16 square, how many different type of genotype and phenotype are there? So 4 different type of phenotype and 9 different type of genotypes are there. 4 phenotype and 9 different type of genotype. Okay? Can you, using the Punnett square data work out? Can you, using the Punnett square data work out the genotypic ratio at the F2 stage and fill in the format given? It is the genotypic ratio also 9:3:3:1. काम करनी है वैसे, इसलिए इन्होंने जल्दी कराया. ठीक है, तो कोई दिक्कत की बात नहीं. नेक्स्ट टॉपिक इज chromosomal theory of inheritance. Mendel published his work on inheritance of character in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, his concept of gene or factor in Mendel's word as a stable and discrete unit that controlled the expression of the traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, through Mendel's work suggested that genes were discrete units. He could not provide any physical proof for the existence of factors or say what they were made of. प्रूव ही नहीं कर पाया. ठीक है. In 1900, 3 scientists, De Vries, Correns and von Teschmark, independently rediscovered Mendel's result on the inheritance of the characters. Also, by this time, due to the advancement in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes. This led to the discovery of a structure in the nucleus that appeared to double and divide just before each cell division. They were called chromosomes, that is colored bodies, as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosome was parallel to the behavior of genes. Behavior of chromosome was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosome during mitosis, that is equational division, and during meiosis, that is reduction division. The important thing to remember are that chromosome as well as genes occur in pair. Chromosome as well as genes occur in pair. The two alleles of a gene pair are located on homologous site on homologous chromosome. The two alleles of a gene pair are located on homologous site on homologous chromosome. ये important line है. The two alleles of a gene pair are located on homologous site on homologous chromosome. ठीक है, डायग्राम दिया हुआ है meiosis and germ cell formation in a cell with 4 chromosomes. Can you see how chromosomes segregate when germ cells are formed? ठीक है. Next is comparison between the behavior of chromosome and genes. कॉलम ए दिया हुआ है यहाँ पे जिसमें occur in pairs. सेकंड जो दिया हुआ है पेयर occur in pair. फर्स्ट वाले में segregate at the time of gamete formation such that only one of the each pair is transmitted to a gamete. सेकंड कॉलम में segregate at gamete formation and only one of the each pair is transmitted to a gamete. यहाँ पे कॉलम जो ए है वो क्रोमोसोम के लिए है एंड कॉलम बी जो है वो जीन के लिए. तो क्रोमोसोम भी ऑक्यूर होता है पेयर में और जीन भी होते हैं पेयर. क्रोमोसोम सेग्रीगेट एट द टाइम ऑफ गैमेट फॉर्मेशन सच दैट ओनली वन ऑफ ईच पेयर इज ट्रांसमिटेड टू ए गैमेट, और जीन्स सेग्रीगेट एट ए गैमेट फॉर्मेशन एंड ओनली वन ऑफ ईच पेयर इज ट्रांसमिटेड टू ए गैमेट. क्रोमोसोम independent pair segregate independently of each other, while in genes one pair segregate independent of the another pair. ठीक है, of the another pair. One pair segregate independently of another pair. ठीक है, ये किसके लिए? जीन के लिए. ठीक है. During anaphase of meiosis first, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosome of 4 different colors in the left and right columns. In the left column, possibility 1, that is orange and green, is segregating together, but in the right hand column, possibility 2, the orange chromosome is segregating with the red chromosome. So the possibility 1 जो दिया हुआ है, 1 long orange and short green chromosome and long yellow and short red chromosome at the same poles. Possibility 2 में दिया हुआ है 1 long orange and short red chromosome and a long yellow and short green chromosome at the same pole. ठीक है, डायग्राम दिया हुआ है यहाँ पे इंडिपेंडेंट असॉर्टमेंट ऑफ द क्रोमोसोम्स. ठीक है. सो नेक्स्ट इज Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton and Boveri argued that the pairing and segregation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with the Mendelian principles and called it as a chromosomal theory of inheritance. किसने यूनाइट किया? Sutton united the knowledge of chromosomal segregation with Mendelian principle and called it as the chromosomal theory of inheritance. ठीक है. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan, that is T.H. Morgan, and his colleagues led to discovering the basis for the variation that sexual reproduction is produced. Following this synthesis of ideas, experimental verification of chromosomal theory of inheritance by T.H. Morgan and his colleague led to the discovering the basis for variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, that is Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. क्या reason है? They could be grown on a simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and the female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscopes. Next is linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by the Mendel in peas. For example, Morgan hybridized yellow-bodied, white-eyed female to brown-bodied, red-eyed male. Morgan hybridized yellow-bodied, white-eyed female to brown-bodied and red-eyed male, लाल आँखों वाला पुरुष, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosome, and so quickly that when the two genes in dihybrid cross were situated on the same chromosome, the proportion of the parental gene combination were much higher than the non-parental type. ठीक है? Proportion of parental gene combination were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage. To describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Again, I am repeating it. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combination. Okay? Morgan and his group also found that even when the genes were grouped on the same chromosome, some genes were very tightly linked, showed very low recombination, that is cross A दिया हुआ फिगर में, while other were loosely linked, showed higher recombination, जो दिया हुआ है फिगर 4.11 में क्रॉस B है. ठीक है? For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between the genes and mapped their position on the chromosome. His student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes. His student Alfred Strutman used the frequency of recombination between the gene pair on the same chromosome. On the same, change करके कुछ भी कर सकता है, but correct है on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genome, as was done in the case of human genome sequencing project described later. ठीक है, important line है. Today genetic maps are extensively used as a starting point in the sequencing of whole genome, as was done in the case of the human genome sequencing project described later. यहाँ पे क्रॉस है और क्रॉस भी दी हुई है, ठीक है, पेरेंटल दिया हुआ है, फन जनरेशन, वैसे इतना इंपॉर्टेंट नहीं है, यहाँ से कुछ आया नहीं, ठीक है. जो फिगर के यहाँ दिया हुआ है लिंकेज, रिजल्ट्स ऑफ टू डायहाइब्रिड क्रॉसेस कंडक्टेड बाय मॉर्गन. क्रॉस ए शोज क्रॉसिंग बिटवीन जीन स्मॉल y and small w. Cross B showing crossing between genes small w and small m. Here, dominant wild type alleles are represented with plus sign in superscript. Note the strength of the linkage between small y and small w is higher than small w and small m. Next is polygenic इनहेरिटेंस. ठीक है, यहाँ से सारे इंपॉर्टेंट टॉपिक हैं, यहाँ से आते हैं भर भर के क्वेश्चन. Mendel's studies mainly describe those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short pupils as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by 3 or more genes and are thus called as polygenic traits. For example, in human, we don't have just tall or short pupil as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by 3 or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into the account the influence of environment. Human skin color is another classic example for this. इसका polygenic inheritance का example है human skin color. ठीक है, in a polygenic trait, the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. To understand this better, let us assume that 3 genes capital A, capital B, capital C control skin color in human, with the dominant forms capital A, capital B and capital C responsible for dark skin color, and the recessive forms small a, small b and small c for light skin color. लाइट स्किन के लिए स्मॉल है और डार्क स्किन के लिए कैपिटल है. ठीक है, द जीनोटाइप विथ ऑल द डोमिनेंट एलील कैपिटल ए, कैपिटल ए, कैपिटल बी, कैपिटल सी will have the darkest skin color, and that with all the recessive alleles small a, small b, small c will have the lightest skin color. As expected, the genotypes with 3 dominant alleles and 3 recessive alleles will have an intermediate skin color. In this manner, as expected, the genotype with 3 dominant recessive the genotype with the 3 dominant alleles and 3 recessive alleles will have an intermediate skin color. In this manner, the number of each type of cells in the genotype would determine the darkness or lightness of the skin in an individual. Next is pleiotropy. We have so far seen the effect of a gene on a single phenotypic or trait. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a pleiotropic gene. However, instances where a single gene exhibits multiple phenotypic expression, एक ही जीन ज़्यादा पावर बता रहा है, ठीक है, नखरे. A single gene exhibit multiple phenotypic expression. Such a gene is called pleiotropic genes. ठीक है, कंफ्यूज हो गया प्लायोट्रोपिक और पॉलीजेनिक में, तो अब क्लियर है कंफ्यूजन, ठीक है. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this is the disease phenylketonuria. An example of this is the disease phenylketonuria, which occurs in human. This disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase, that is single gene mutation. This disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase, that is single gene mutation. This manifests itself through phenotypic expression characterized by the mental retardation and reduction in hair and skin pigmentation. This manifests itself through phenotypic expression characterized by mental retardation and reduction in hair and skin pigmentation. क्या क्या करता है? Mental retardation and reduction in hair and the skin pigmentation. Next is sex determination. The mechanism of sex determination has always been a puzzle before the genetist. The initial clue about the genetic chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in the insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetical the cytological observation made in the number of insects led to the development of the concept of genetic chromosomal basis of sex determination. Hanking in 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave a name to this structure as the X body, but he could not explain its significance. Further investigation by other scientists led to the conclusion that the X body of the Hanking was in fact a chromosome. ठीक है, हैंकिंग उसे प्रूव नहीं कर पाया उसके सिग्निफिकेंस को, ठीक है, किसकी एक्स बॉडी, but could not provide his explain his significance. Further investigation by other scientists led to the conclusion that X body of Hanking was in fact a chromosome, and that is why it was given the name X chromosome. ठीक है, it was also observed that in a large number of insects, the mechanism of sex determination is of the XO type. That is, all X bear an additional X chromosome besides the other chromosomes autosomes. On the other hand, some of the sperms bear the X chromosome where the some do not. ठीक है, that is, all X bear an additional X chromosome besides the other chromosome, beside the other chromosomes, that is autosomes. On the other hand, some of the sperms bear the X chromosome whereas some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome becomes male. Do you think the number of chromosomes in the male and female are equal? Of course. Due to the involvement of the X chromosomes in the determination of sex, it was designated to be the sex chromosomes. It was designed to be the sex. It was designed to be the sex chromosome. and the rest of the chromosome were named as autosomes. Grasshopper is an example of XO type of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosome. Females have a pair of X chromosome. This observation led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, XY type of sex determination is seen where both male and female have same number of chromosomes. XY type of sex determination is seen where both male and female have same number of chromosomes. Clear? Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosomes. Both males and females bear same number of autosomes. Both males and females bear same number of autosomes. Hence, the males have autosomes plus XY, while females have autosomes plus XX. In human beings and in Drosophila, the male have one X and one Y chromosome. In human being and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosome besides autosomes. Here the figure is given. In the figure there is a determination of sex by chromosomal differences. Picture A and B both in humans and in Drosophila, the female has a pair of XX chromosomes, that is homogametic, and the male is XY, that is heterogametic composition. ठीक है? और C वाला जो फिगर दिया हुआ उसमें in many birds female has a pair of dissimilar chromosome ZW and male two similar ZZ chromosome. बर्ड्स में ZZ सिमिलर क्रोमोसोम मेल के लिए होता है और ZW डिसिमिलर फीमेल के लिए होता है. ठीक है? फीमेल में डिसिमिलर पेयर होता है. क्लियर? अंडरस्टूड? सो लेट्स प्रोसीड फर्दर. In the above description, you have studied about two types of sex determination mechanism, that is XO type and XY type. But in both cases, males produce two different types of gamete, either with or without X chromosome, or second is some gametes with X chromosome and some with Y chromosome. Such types of sex determination mechanism is designated to be the example of male heterogamety. Be the example of कौन सा? Male heterogamety का. ठीक है? In some other organisms, example birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both male and female, but two different types of gametes in terms of the sex chromosomes are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to be the Z and W chromosomes. In this organism, the female have one Z and one W chromosome, whereas males have a pair of Z chromosome besides the autosomes. Clear? Next is sex determination in humans. It has already been mentioned that the sex determining mechanism in case of humans is XY type. Out of 23 pairs of chromosome presents, 22 pairs are exactly same in both male and females. These are the autosomes. Clear? A pair of X chromosome are present in the female, whereas the presence of an X and Y chromosome are determinant of the male characteristic. Clear? During spermatogenesis among male, two types of gametes are produced. That is, 50% of the total sperm produced carry the X chromosome and the rest 50% has Y chromosome. Besides the autosomes. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case the ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female XX, and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. There is always 50% probability of either a male or a female child. It is unfortunate that in our society, women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. Clear? So the next is sex determination in human. में पढ़ लिया है अपन ने, अब नेक्स्ट पढ़ेंगे सेक्स डिटर्मिनेशन इन हनी बी. ठीक है. द सेक्स डिटर्मिनेशन इन हनी बी इज बेस्ड ऑन द नंबर ऑफ सेट्स ऑफ क्रोमोसोम an individual receives. An offspring formed from the union of a sperm and an egg develops as a female, that is queen or worker. ठीक है. Offspring formed from the union of a sperm and egg develops as a female, that is queen or worker, and an unfertilized egg develops as a male. Unfertilized egg develop होता है male, जिसको ड्रोन कहते हैं. ठीक है. By means of parthenogenesis. ठीक है. This means that the males have half the number of chromosomes than that of a female. The females are diploid having 32 chromosomes and the males are haploid, that is having 16 chromosomes. Males में 16 chromosome होते हैं, females में 32 chromosome होते हैं. This is called as haplodiploid sex determination system and has a special characteristic feature such as the male produce sperms by mitosis. They do not have father and thus cannot have sons, but have a grandfather and can have grandsons. ठीक है, ये important line है. ठीक है, and has a special characteristic feature such as the males produce sperm by mitosis. They do not have father and thus cannot have sons, but have a grandfather and can have grandsons. ठीक है. यहाँ पे डायग्राम भी दिया हुआ है सेक्स डिटर्मिनेशन इन हनी बी. ठीक है. सो द नेक्स्ट इज हाउ इज द सेक्स डिटर्मिनेशन मैकेनिज्म डिफरेंट इन द बर्ड्स. इज द स्पर्म और द एग रिस्पॉन्सिबल फॉर द सेक्स ऑफ द चिक्स. क्या क्वेश्चन दिया हुआ है, अगेन आई एम रिपीटिंग इट. हाउ इज द सेक्स डिटर्मिनेशन मैकेनिज्म डिफरेंट इन द बर्ड्स. इज द स्पर्म और द एग रिस्पॉन्सिबल फॉर द सेक्स ऑफ द चिक्स. The sex of the chick is determined by the egg, not the sperm. This is different from human. Clear? Only the egg is responsible for the sex of the chicks. ठीक है? This is the answer of that question. So the next is mutation. Mutation is a phenomenon which results in alteration of DNA sequence and consequently results in change in the genotype and the phenotype of an organism, and consequently results in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. As you will learn in chapter 5, one DNA helix runs continuously from one end to the other in each chromatid in a highly supercoiled form. Therefore, loss, that is deletions, or gain, that is insertion or duplication of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on genes are known to be located on chromosomes, alternation in chromosome results in abnormality or aberrations. Clear? Genes are known to be located on chromosomes. Alternation in chromosome results in abnormality or aberrations. Chromosomal aberrations are commonly observed in cancer cell. कौन से सेल में ऑब्जर्व होती है? कॉमनली कैंसर सेल्स. In addition to the above, mutation also arises due to change in a single base pair of DNA. This is known as point mutation. A classical example of such a mutation is sickle cell anemia. किसका एग्जाम्पल है? पॉइंट म्यूटेशन का. Deletion and insertion of a base pair of DNA cause frame shift mutations. You will see this in chapter 5. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemicals and physical factors that induce mutations. These are referred to as mutagens. UV radiation can cause mutation in organism. It is a mutagen. Ultraviolet radiation, that is UV radiation, can cause mutation in organism. It is a mutagen. ठीक है. अब यहाँ पे नेक्स्ट टॉपिक है जेनेटिक डिसऑर्डर्स. नेक्स्ट इज पेडिग्री एनालिसिस. The idea that orders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of trait in human beings began. Since it is evident that control crosses that can be performed in pea plant or some other organism are not possible in case of human beings, a study of the family history about inheritance of a particular trait provides an alternative. Since it is evident that control crosses that can be performed in pea plant or some other organism are not possible in case of human being, a study of the family history about inheritance of a particular trait provides an alternative. ठीक है. Such an analysis of traits in several of generation of a family is called as the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generation. यहाँ पर डायग्राम दिया हुआ है, सिम्बॉल्स यूज्ड इन द फीमेल पेडिग्री एनालिसिस. सबसे फर्स्ट है मेल, जैसे स्क्वायर से डिनोट करते हैं, सर्कल को फीमेल से डिनोट करते हैं, और जो स्क्वायर है वो खड़ा है, मीन्स आधा इधर बीच वन पॉइंट पर खड़ा हुआ, उसे कहते हैं सेक्स अनस्पेसिफाइड. बॉक्स है रेड कलर का स्क्वायर, और फिर सर्कल है रेड कलर का, फिर वो सेक्स अनस्पेसिफाइड वाला रेड कलर में, वो उस सिंबल तीनों साथ में है, तो उन्हें कहते हैं अफेक्टेड इंडिविजुअल. एक स्क्वायर है और एक सर्कल है, दोनों कनेक्ट है सिंगल लाइन से, तो उसे बोलते हैं मैटिंग. बॉक्स है स्क्वायर का और डबल लाइन से कनेक्टेड है सर्कल से, तो उसे कहते हैं मैटिंग बिटवीन रिलेटिव्स, दैट इज कॉन्सेजीनियस मैटिंग. बना रहे हो दिमाग में ये डायग्राम्स? आ रहा है क्लियर? ठीक है. स्क्वायर है, स्क्वायर कनेक्ट है सर्कल से, और उन दोनों को कनेक्ट करके नीचे उनकी प्रोजेनी बनाई है, एक सर्कल और एक स्क्वायर, तो उसे कहते हैं पेरेंट्स अबव एंड चिल्ड्रन बिलो इन ऑर्डर ऑफ बर्थ, दैट इज लेफ्ट टू राइट. ऑर्डर में बताया हुआ है लेफ्ट से राइट. फिर नेक्स्ट है स्क्वायर, उसे कनेक्ट किया है सर्कल से, और नीचे है स्क्वायर रेड कलर में अफेक्टेड, मीन्स इसका मतलब है पेरेंट्स विथ मेल चाइल्ड अफेक्टेड विथ डिसीज. फिर नेक्स्ट है जो सेक्स अनस्पेसिफाइड वाला था स्क्वायर, जो वन एंगल पे खड़ा हुआ है बॉक्स, ठीक है, और उसके अंदर 5 है, मीन्स उसका मतलब है 5 अनअफेक्टेड ऑफ स्प्रिंग. ठीक है, उसका मतलब क्या हुआ? 5 अनअफेक्टेड ऑफ स्प्रिंग. क्लियर? इन ह्यूमन जेनेटिक्स, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or diseases. ठीक है, in human genetic pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait and abnormality or diseases. Some of the important standard symbols used in the pedigree analysis have been shown in the figure 4.13, which we have already discussed. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA present in the chromosome. Clear? DNA is the carrier of genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, changes or alteration do takes place occasionally. Such an alteration or changes in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. ठीक है, नेक्स्ट इज मेंडेलियन डिसऑर्डर्स. यहाँ से भी क्वेश्चन आते हैं भर के. क्लियर? ब्रॉडली genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation in the single gene. ठीक है, Mendelian disorders are mainly determined by alteration or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern inheritance of such Mendelian disorder can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome as in case of hemophilia. It is evident that these X-linked recessive traits show transmission from carrier female to male progeny. It is evident that the X-linked recessive trait shows transmission from carrier female to the male progeny. A representative pedigree is shown in the figure 4.14 for dominant and recessive traits. Discuss with your teacher and design the pedigree for characters linked to both autosomes and sex chromosome. यहाँ पे दिया हुआ है, ठीक है, रिप्रेजेंटेटिव पेडिग्री एनालिसिस दी हुई है, ठीक है, जिसमें फर्स्ट दी हुई है ऑटोसोमल डोमिनेंट ट्रेट. ऑटोसोमल डोमिनेंट ट्रेट का एग्जांपल है मायोटोनिक डिस्ट्रॉफी, और सेकंड दी हुई है ऑटोसोमल रेसेसिव डिसऑर्डर, that is autosomal recessive trait. For example, sickle cell anemia is the recessive trait, and myotonic dystrophy is autosomal dominant trait, or example myotonic dystrophy. Clear? Next is color blindness. It is a sex-linked recessive disorders due to defect in either red or green color. It is a sex-linked recessive disorder due to defect in either red or green cone of eyes, resulting in failure to discriminate between red and green color. रेड और ग्रीन की पता नहीं चलती, ठीक है। दिस डिफेक्ट इज ड्यू टू द म्यूटेशन इन सर्टेन जीन्स प्रेजेंट इन द एक्स क्रोमोसोम. It occurs in about 7% of the males and only about 0.4% of females. This is because genes that leads, again I am repeating it, the defect is due to a mutation in certain genes present in the X chromosome. It occurs in about 8% of the males and only 0.4% of females. This is because the genes that lead to red-green color blindness are on the sex chromosome, that is X chromosome. Males have only one X chromosome and the females have two. The son of a woman who carries the gene has 50% chances of being colorblind. The son of a woman who carries the gene of color blindness has a 50% chances of being colorblind. ठीक है. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and father is colorblind. ठीक है. Daughter will not normally be colorblind unless her mother is carrier and her father is colorblind. So the next is hemophilia. यहाँ से भी क्वेश्चन आता ही आता है, श्योर पक्का. Next is hemophilia. The sex-linked recessive disease. Sex-linked recessive disease है hemophilia, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. In this disease, a single protein that is a part of a cascade of protein involved in the blood clotting or protein is affected. A single protein that is a part of the cascade of protein involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female carrier for hemophilic. Due to this, in affected individual, a simple cut will result in non-stop bleeding. The heterozygous female carrier for hemophilia may transmit its disease to sons. The possibility of a female becoming hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic. The possibility of female becoming a hemophilic is extremely rare because mother of such female has to be at least carrier and the quarter. ठीक है. Should be. The possibility of a female becoming hemophilic is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic, unviable the later stage. The family pedigree of Queen Victoria shows a number of hemophilic descendants. She was carrier for disease. Clear? The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of this disease. Next is sickle cell anemia. Sickle cell anemia, ठीक है. This is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene or heterozygous. Clear? This is an autosome-linked recessive trait. कौन? sickle cell anemia autosomal linked recessive trait है, ठीक है. That can be transmitted from Parents to the offspring when the partners are carrier for the gene or heterozygous. Disease is controlled by a single pair of allele HBA and HBS. Out of 3 possible genotypes, only homozygous individuals for HBS shows the deceased phenotype. A heterozygous HB की power capital A, HB की power is capital S. Individuals appear apparently unaffected, but they are carrier of the diseases, as 50% of probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell anemia trait. Next is the effect is caused by the substitution of glutamic acid by valine. Glutamic acid glue by valine. क्लियर? यहाँ पे दिया हुआ है valine at the 6th position of the beta globin chain of the human globin molecule. ये लोकेशन दी हुई है, ठीक है? यहाँ पे डायग्राम दिया हुआ नॉर्मल जीन का, जिसमें माइक्रोग्राफ ऑफ द RBC and the amino acid composition of the relevant portion beta chain of hemoglobin. First is from a normal individual. Second diagram B is from an individual with sickle cell anemia. Clear? So the next is the substitution of amino acid in the globin protein results due to a single base substitution at the 6th codon of the beta globin chain from GAG to GUG. Gug se gug. The mutant hemoglobin molecule undergo polymerization under low oxygen tension. Causing the changes in the shape of the RBCs from biconcave disc to elongated sickle-like structure. The mutant hemoglobin molecule undergo polymerization under low oxygen tension. इसकी presence में under low oxygen tension, causing the changes in the structure in the shape of RBC from biconcave disc to elongated sickle-like structure. Next is phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual, ठीक है? Affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of this in brain results in mental retardation. These are also excreted through urine because its poor secretion by kidney. Accumulation of this in brain result in mental retardation. Next is thalassemia. This is also an autosome-linked recessive blood disease transmitted from parents to the offspring when both the parents are unaffected, carrier for the gene, that is, or heterozygous. The effect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chain, that is, alpha and beta chains that make up hemoglobin. This causes the formation of abnormal hemoglobin molecule, resulting into anemia, which is characterized of the disease. चलो, again I am repeating the thalassemia. Listen it properly, ठीक है? This is also an autosome-linked recessive blood disease. Autosome-linked recessive blood disease transmitted from parent to the offspring when both the parents are unaffected, carrier for the gene, that is, or heterozygous. The defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chain, that is alpha and beta chains that make up hemoglobin. This causes the formation of abnormal hemoglobin molecule resulting into anemia, which is characteristic of the disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. Thalassemia is classified according to which chain of the hemoglobin is affected. कौन सी चेन अफेक्ट हुई है, ठीक है. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Alpha chain, that is alpha thalassemia. Alpha thalassemia is controlled by two closely linked genes, that is HbA1 and HbA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. Again I am repeating it. Chalo. Alpha thalassemia is controlled by the two closely linked genes HbA1 and HbA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin chains produced. यह आ सकता है लाइन ऐसी की ऐसी चेंज होगी कुछ भी. The more genes affected, the more alpha globin molecule produced. It's wrong. It's false statement. Correct statement is the more genes affected, the less alpha globin chains produced. It is correct. Next, proceed further. While beta thalassemia is controlled by a single gene HBB on chromosome 11 of each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is quantitative problem of synthesizing too few globin molecule, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin chain. सिकल सेल एनीमिया है वो क्वालिटेटिव प्रॉब्लम है, मीन्स क्वालिटी दिखाता है सिकल सेल एनीमिया, और थैलेसेमिया है वो क्वांटिटेटिव प्रॉब्लम ऑफ सिंथेसाइजिंग टू फ्यू ग्लोबिन मॉलिक्यूल. ठीक है, दोनों में डिफरेंस क्लियर है. अगेन आई एम रिपीटिंग इट. अल्फा थैलेसेमिया है वो इज कंट्रोल्ड बाय टू क्लोजली लिंक्ड जीन HbA1 एंड HbA2 ऑन क्रोमोसोम 16, और जो बीटा थैलेसेमिया है is controlled by a gene HBB on chromosome 11 of each parent. ठीक है, अल्फा 1 का ताल्लुक है क्रोमोसोम 16 से, मीन्स अल्फा थैलेसेमिया का. ठीक है, और बीटा थैलेसेमिया का ताल्लुक है HBB ऑन क्रोमोसोम 11 से. क्लियर हो गया, माइंड में सेट, बिल्कुल आराम से चल रहा है. ठीक है. नेक्स्ट इज क्रोमोसोमल डिसऑर्डर्स. The chromosomal disorders, on the other hand, are caused due to absence or excess or abnormal arrangement of one or more chromosomes, failure of segregation of chromatids during cell division. Failure of segregation of chromatids during cell division cycle results in the gain or loss of a chromosome called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. Down syndrome results in the gain of extra copy of chromosome 21. Similarly, Turner's syndrome results due to loss of an X chromosome in human females. Similarly, Turner syndrome results due to loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomena is known as polyploidy. Failure of cytokinesis after telophase stage of cell division results in an increase in whole set of a chromosome in an organism, and this phenomena is known as polyploidy. This condition is often seen in plants. किसमें सीन होती है? ऑफन प्लांट्स में. नेक्स्ट इज द टोटल नंबर ऑफ क्रोमोसोम इन ए नॉर्मल ह्यूमन सेल इज 46, that is 23 pairs. Out of this, 22 pairs are autosomes and 1 pair of chromosome are sex chromosome. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any of. Again I am repeating it. Sometimes, through rarely, either an additional copy of a chromosome may be included in an individual, or an individual may lack one of any of one pair of chromosome. Individual may lack one of any one pair of chromosomes. This situation are known as trisomy or monosomy of a chromosome respectively. Trisomy किसे कहते हैं? Either an additional copy of chromosome may be included in an individual, इसे कहते हैं trisomy. और monosomy कहते हैं either an individual may lack one of any one pair of chromosome. Correct? Clear? Such a situation leads to a very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of chromosomal disorder. Down syndrome, Turner syndrome, Klinefelter syndrome are common example of chromosomal disorders. ठीक है? Clear? Now we are going to read Down syndrome. The cause of this genetic disorder is the presence of an additional copy of chromosome number 21. कौन सा chromosome number 21? This disorder was first described by Langdon Down in 1866. The affected individual is short-statured with a small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor, and mental development is retarded. Again I am repeating it. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, that is trisomy of 21. This disorder was first described by Langdon Down in 1866. The affected individual is short-statured with a small round head, furrowed tongue, and partially open mouth. क्या क्या? Affected individual is short-statured with a small round head, furrowed tongue, and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor, and mental development is retarded. Clear? ये हो गया डाउन सिंड्रोम. इतना स्लो चल रही है कि आपको कुछ भी करने की ज़रूरत नहीं है, बस ध्यान से सुनो. ठीक है? ऑटोमेटिक सेट हो जाएगा दिमाग. यहाँ पे डायग्राम में दिया हुआ है रिप्रेजेंटेटिव फिगर शोइंग एन इंडिविजुअल इन्फ्लिक्टेड विद डाउन सिंड्रोम एंड द करेस्पॉन्डिंग क्रोमोसोम ऑफ एन इंडिविजुअल. फिगर में जो दिया हुआ है यहाँ पे भी सिम्टम्स दिए हुए हैं, ऐसे भी पूछ सकता है फिगर से. ठीक है? डाउन सिंड्रोम का यहाँ पे दिया हुआ है फ्लैट बैक ऑफ हेड, मैनी लूप्स ऑन फिंगर टिप्स, palm crease, broad flat face, big and wrinkled tongue, congenital heart disease. ये है डाउन सिंड्रोम. ठीक है? नेक्स्ट वी आर गोइंग टू रीड Klinefelter syndrome. This genetic disorder is also caused due to presence of an additional copy of X chromosome resulting in a karyotype of 47 XXY. Such an individual has overall masculine development. However, the feminine development, that is development of breast, is known as gynecomastia, is also expressed. Such individuals are sterile. Again I am repeating it. This genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting in the karyotype of 47, that is XXY. Such an individual has overall masculine development. However, the feminine development, that is development of breast, gynecomastia, is also expressed. Such individuals are sterile. ठीक है? Next is Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosome. Such a disorder is caused due to the absence of one of the X chromosome, that is 45 with XO. Such females are sterile with rudimentary ovaries. Such females are sterile as ovaries are rudimentary, besides other features including lack of secondary sexual character. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual character. यहाँ पे डायग्रामेटिक रिप्रेजेंटेशन दिया हुआ है जेनेटिक डिसऑर्डर ड्यू टू सेक्स क्रोमोसोम कंपोजिशन इन ह्यूमंस. इसमें क्लीन फेल्टर दिया हुआ है, ठीक है, जिसमें यहाँ पे डायग्राम में दिया हुआ है टॉल स्टेचर विथ फेमिनाइज्ड कैरेक्टर. किस में आएगा? क्लीन फेल्टर सिंड्रोम में. और शॉर्ट स्टेचर एंड अंडर डेवलप्ड फेमिनिन कैरेक्टर आएगा टर्नर सिंड्रोम में. टॉल स्टेचर विथ फेमिनिन कैरेक्टर है क्लीन फेल्टर सिंड्रोम में, और शॉर्ट स्टेचर एंड अंडर डेवलप्ड फेमिनिन कैरेक्टर है टर्नर सिंड्रोम. क्लियर? नेक्स्ट इज समरी. चलो जल्दी जल्दी पढ़ता हूँ. जेनेटिक इज अ ब्रांच ऑफ बायोलॉजी विच डील्स विद द प्रिंसिपल ऑफ इनहेरिटेंस and its practices. Progeny resembling the parents in morphological and physiological features has attracted the attention of many biologists. Mendel was the first to study the phenomenon systematically. While studying the pattern of inheritance in pea plants of contrasting character, Mendel proposed the principle of inheritance, which are today refers to as Mendel's law of inheritance. He proposed that the factors, later named as genes, regulating the character are found in pair known as allele. He observed that the expression of the character in the offspring follow a definite pattern in different first generation F1, second, F2 and so on. Some characters are dominant over others. The dominant characters are expressed when factors are in heterozygous condition, that is law of dominance. Dominant character are expressed when the factors are in heterozygous condition. ठीक है. The recessive character are only expressed in homozygous condition. Dominant heterozygous condition में होते हैं और recessive homozygous में होते हैं. Homozygous condition में क्या होते हैं? expressed. ठीक है. The character never blend in heterozygous condition. A recessive character that was not expressed in heterozygous condition may be expressed again when it becomes homozygous. As characters segregate while formation of gametes, that is law of segregation. Not all characters show true dominance. Some characters show incomplete and some show codominance. When Mendel studied the inheritance of two characters together, it was found that the factor independently assort and combine in all permutations and combinations, that is law of independent assortment. Different combination of gametes are theoretically represented in a square tabular form known as Punnett square. The factor now known as gene on chromosome regulating the character are called the genotype, and the physical expression of the character is called the phenotype. After knowing that the genes are located on the chromosome, a good correlation was drawn between Mendel's law, segregation and assortment of chromosomes during meiosis. The Mendel's law were extended in the form of chromosomal theory of inheritance. Later it was found that Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosome. These genes were called as linked genes. Closely located genes assorted together and distantly located genes due to recombination assorted independently. Linkage maps therefore correspond to arrangement of genes on a chromosome. Many genes were linked to sex. Again I am repeating it slowly. Different combination of gametes are theoretically represented in a square tabular form known as Punnett square. The factor now known as genes on chromosome regulating the character are known as the genotype, and the physical expression of the character is called the phenotype. After knowing that the genes are located on the chromosome, a good correlation was drawn between Mendel's law segregation and assortment of chromosome during meiosis. The Mendel's law were extended in the form of chromosomal theory of inheritance. Later, it was found that the Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosome. These genes were called as linked genes. Closely located genes assorted together and distantly located genes due to recombination assorted independently. Linkage maps therefore correspond to arrangement of genes on a chromosome. Many genes were linked to sexes also and called as sex-linked genes. The two sexes, male and female, were found to have a set of chromosomes which were common and another set which was different. The chromosomes which were different in two sexes were named as sex chromosomes. The remaining set was named as autosomes. In humans, a normal female has 22 pairs of chromosomes and a pair of sex chromosomes, that is XX. A male has 22 pairs of autosomes and a pair of sex chromosomes XY. The remaining set was named as autosomes. In human, a normal female has 22 pairs of autosomes and a pair of sex chromosomes XX. A male has 22 pairs of autosomes and a pair of sex chromosomes XY. In chicken, sex chromosomes in males are ZZ and in females are ZW. Chickens में कौन से होंगे? Sex chromosome in male are ZZ, that is homogamitic, and in female are ZW, that is heterogamitic. Mutation is defined as changes in the genetic material. A point mutation is change of a single base pair in DNA. Sickle cell anemia is caused due to change of one base in the gene coding for beta chain of hemoglobin. Inheritable mutations can be studied by generating a pedigree of a family. Some mutation involves changes in all set of a chromosome, that is polyploidy, or change in a subset of chromosome number, that is aneuploidy. This helped in understanding the mutational basis of genetic disorders. Down syndrome is due to trisomy of chromosome 21, where there is an extra copy of chromosome 21 and consequently the total number of chromosomes becomes 47. In Turner syndrome, one X chromosome is missing and the sex chromosome is as XO. And in Klinefelter syndrome, the condition is XXY. This can be easily studied by analysis of karyotopes. In Turner syndrome, one X chromosome is missing. And Down syndrome is due to trisomy of chromosome 21, where there is an extra extra copy of chromosome 21 and consequently the total number of chromosomes becomes 47. With this we completed the chapter. Thank you for hearing.